that switch is definitely there.
Thank you. And our last talk for the session is from Dr. Jack Rogers on mapping of cardiac propagation and deformation.
All right, thanks, Nanad, and thanks, Joel J. and GQ for the invitation and organizing this. So I work in the cardiac rhythm management lab over across the parkway, the parking lot. So we have a large animal lab, and we do a lot of instrumentation development, a lot of cardiac mapping, so that's what I'll mostly talk about today. So I think there's a lot of appreciation these days that the regenerative therapies have a lot of potential for inducing arrhythmias. This is some data from monkeys that got cell therapy, and you can see that there's lots of VT that occurs in these animals, which is not good. So long story short, cardiac waves don't like heterogeneity. So, you know, if you have some kind of a therapy that's doing something that's going to give you either absent or imperfect coupling or heterogeneous electrical properties, wavefronts have a lot of potential to fragment and break, re-entry conform. You can also get inappropriate automaticity, you know, triggers that can also cause arrhythmias from other mechanisms. So mapping is a tool that can help to diagnose that. So this is kind of the basic scheme for optical mapping techniques, at least for membrane potential mapping. Normally isolated perfused hearts are used. They're stained with a potentiometric dye. Dye 4NF is kind of the common one. Then you excite the tissue with a light that's kind of on this steep flank of the absorption spectrum, and then record on one side of the emission spectrum. When the tissue depolarizes, both of these spectra shift to shorter wavelengths, so you're shifting the spectrum out from underneath your kind of fixed excitation and emission bandwidths, and with this particular setup, that will cause small negative deflections that are proportional to the membrane potential. So this is kind of one setup that we built for this. This was put together so we could image the entire surface of large animal hearts simultaneously. So the basic idea is that we take a heart out, we put it in the middle of the perfusion system here. We have 4 mapping cameras. So these are fast sensitive video cameras appropriate for optical mapping that surround the heart, so we can image it from 4 different directions. Then we have another camera that spins around the heart. It takes a bunch of images as it spins around, and from that we can build up a model of the epicardial surface of the heart, which is kind of what's shown here. Once you have that model, you can take the data from the movies from the 4 mapping cameras and paste it all together and get a continuous data set showing the whole surface. So this is a pig heart fibrillating. The blue is where the tissue is resting. The kind of orange-yellow blobs or fibrillation wavefronts circulating around. So that's kind of the data that we can get with this. This is another view from that instrument. So now what we have is a pig heart that was panoramically mapped. It's been unrolled with a map projection, so you can see the whole epicardium. And what we did, we started over here. We gave it an electrical stimulus that started 2 counter-rotating rotors, so that this is activating the heart at a high rate. And what happens over here, this is where the 2 ventricles come together on the anterior side. And just that heterogeneity over there, where the muscle fibers come together from the 2 ventricles, that causes get that to start to play. There we go. So there you can see the rotors on the right. The blue are excitation wavefronts, and they're chasing the gray fronts that they're chasing are the repolarization wavefronts. So as the waves kind of bunch up right at the RV insertions, what happens, I think now, is the waves will break. Sometimes they'll heal. Sometimes they stay broken. And when they stay broken, that starts a new rotor. and the whole thing falls apart and it goes right back into ventricular fibrillation. So this is a normal heart, but just kind of the native heterogeneity is enough to throw a relatively simple rhythm back into VF, which is much more complicated and worse. So in that setup, we had to give a drug to the heart that prevents it from contracting, so that the electrical activity is more or less preserved, but it doesn't contract anymore. We would like to be able to map in beating hearts. One reason we would like to be able to study electromechanical interactions between the electrical wave and either stretch that's imposed or stretch that occurs. If you can map in beating hearts, that also gives you the potential to do optical mapping in vivo preparations as well. So the problem with motion artifact has been kind of a long-standing limitation in optical mapping. So we've addressed it with a combination of motion tracking and ratiometry. There we go. So what we do, this is an isolated swine heart. We glue markers to the surface of the heart, and then we can track the motion of the markers. And then for any piece of tissue that's within a triangle of markers, we can interpolate and follow the motion of all of the tissue as the heart beats. So by doing that, we can get rid of a lot of motion artifact. This is a signal from chasing. It's not one pixel. It's chasing one tissue site around. And you can see there's still a lot of motion artifact, but you can see depolarizations here. So it's better, but it's still not where we need to be. There we go. So we need more help, and what we do is to implement a ratiometric technique on top of the motion tracking. So the basic idea with that is that what we do on alternating camera frames, we switch the color of excitation lights. So on one frame, we excite the tissue with this blue color. On the next frame, we excite with cyan, and then we record with a single camera with a single band. So the fluorescence that's elicited by this wavelength is not voltage sensitive, but the fluorescence that's elicited by this color is voltage sensitive. So this is kind of the signal that you get where the blue and cyan elicited fluorescence is interlaced. We uninterlace it by taking alternate frames, and we get these two signals. So there's still lots of motion artifacts here, but you take the ratio, and then you can reconstruct membrane potential. So now that we're tracking motion, we can also measure the deformation of the tissue almost for free. So we add a few extra cameras. Now we can track the markers in 3 dimensions, and we can compute epicardial strains. So this you wouldn't think this would be this hard to start a movie. There we go. Okay, so what you're seeing on the right, these are strains. So the little blue bars, when those are long, the tissue is contracting, and the orientation tells you the principal direction of the contraction. Over here is the electrical map. So the light blue wave going by is a depolarization wavefront, and then it goes through yellow and then red as it plateaus and recovers. So that's the kind of data we can get. What we did in this study, this is using the same instrument. What we've done here, the LAD runs right along this top edge of the mapping array, and we ligated it right about here. So you can kind of see, I don't know if you can see it or not, but there's kind of a faint line here, so that's the border zone. Over here now, you're seeing the strains, and there we go. So in the ischemic region over here, those red lines, that's telling you that the tissue is stretching, so it's bulging out, whereas over here in the more normal tissue, you're still getting contraction, so there's a lot of stretch right here. And what you see there, that is an ectopic beat that's arising right at the border zone where it's highly stretched. So what we're looking for here is to see if early arrhythmias in the first hour or so after an acute occlusion, if those ectopic beats that arise are related to stretch. That one was. In general, they're probably not. We're still working on that data. With an optical mapping, you get high spatial resolution. You can see membrane potential. But if you have a heart, say, that has had a previous open chest procedure, or you've given it some, put a patch on it or given it cells, there's a good chance you're going to have a lot of scarring and adhesions on the tissue, which makes it hard to map optically. So we also have a 528 channel mapping system, electrical mapping system that's, I guess it's older than most of my undergraduate students. So with a system like that, you can put arrays of electrodes on the tissue, usually lower resolution, but you have a better chance of getting signals in hearts that are compromised. So this is what you see during pacing, right? The pacing stimulus, activation, and the T wave tells you when recovery is. So the time between excitation and recovery is of great interest in arrhythmias. Unfortunately, when the tissue is going fast, right, here's a signal taken during VF, the T waves and the QRS kind of fuse together, so you lose that recovery information. So that's kind of a disadvantage of electrical mapping. But here's kind of one type of electrode array that we've done. So we have an array of electrodes on the surface, and then we have an orthogonal kind of cutting plane. So this is a row, like a fence row of needles that was inserted into the tissue. That's what the needles look like. They're about half a millimeter in diameter. So this shows this array here and this array here. That's the epicardial data. The little red lights flash when the tissue is in the process of activating. And this is the data from the transmural needle array. So you can see if you look carefully, there's a rotor over here going around. What we were looking for in this study was whether we could find intramural rotors in this cutting plane and during fibrillation, and we could not. Here's another example of 3D intramural data that you can do with electrical mapping. Here we had an array of a 9x9 array, and with 2 millimeter spacing in both directions. Then we put needles down through this template. So it basically gave us 6 kind of a stacked plane. And this shows you the kind of data that we got from that. This is during a long duration ventricular fibrillation. When the tissue is kind of slowing down, you can see that it's activating first, kind of down endocardially, then propagates up to the surface. So I'm going to stop there. Just wanted to give you some examples of some of the mapping that we do. These are the people that were primarily involved in this work and funding sources. So thank you.
Time for one quick question, and we go to a quick break.
What is the resolution for the stream mapping that you were doing? What does a pixel represent in terms of area?
So the way we're doing it now, we get one strain measurement for each one of those triangles. So the triangles, about 8 millimeters apart is how we try to place them. So we've done some studies that show that the strain within that triangle is pretty much homogeneous. So another thing we could do would be to fit a finite element to that and make it continuous, but we haven't done that yet.
Okay. Thank you.